Hi, and welcome everyone to the webinar series Topics at the Nexus of Climate Change, Fisheries and Blue Foods. This webinar series aims to highlight the impacts of climate change on fisheries, aquaculture, and the communities who depend on them. This webinar series is jointly hosted by the UN Ocean Decade Programs, Blue Food Futures, Fisheries Strategies for Changing Oceans and Resilient Economies, FishSCORE, Sustainability, Predictability, and Resilience of Marine Ecosystems, SUPREME, Sustainability of Marine Ecosystem through Global Knowledge Networks, or SmartNet, and the Fisheries and Marine Ecosystem Model Intercomparison Project, FishMIP, and also the endorsed project, Basin Scale Events to Coastal Impacts, or BECI. Claire has posted a link with more information about all of the different hosting programs. So this month, we're focusing on the topic climate mitigation, fish, and the ocean carbon cycle. We are hosting two speakers. The first one is Keith Rodgers, who will be presenting on the high value of climate mitigation in limiting global fish biomass losses to the year 2300, and Gaël Mariani on the role of fish in the ocean carbon cycle in a changing climate and fishing context. Each presentation will be followed by a question session, and please use the Q&A box to ask your questions. The Q&A box can be found under the More button at the bottom of your screen. So Keith Rodgers, our first presenter, is a principal investigator at the Advanced Institute for Marine Ecosystem Change, or AIMEC, at Tohoku University in Japan, where he has worked since 2024. Together with Daniele Bianchi from UCLA, Keith co-leads the FishMIP working group on projections out of 2300. And today he'll be presenting some of the working group's findings. Over to you, Keith. You have 20 minutes.
Can you hear me? Am I unmuted?
Yes.
So great. Okay, so I will begin by just saying thank you for welcoming this talk, and I'm happy to share with you some results. You can see that this is a climate change focused global scale study, and I didn't list all the collaborators here, but it does include a number of FishMIP contributors with FishMIP models. And the principal collaborator with whom this project started is Daniele Bianchi. So if we could go to the next slide, please. Okay, so just as a matter of scientific background, I wanted to just give you a sense of how this project developed. So I come from more of an Earth system modeling type of that type of work, and so there I've been focused on mechanisms that drive lower trophic level. Until recently, this has been my primary focus: mechanisms controlling lower trophic level responses to climate change. And this work with lower trophic levels and its extension to fish grows out of a collaboration with Daniele Bianchi through our Nature publication from 2 years ago. And there we were focused on understanding uncertainties with mechanisms that can modulate future NPP in Earth system model projections to 2300. So these kind of extended well beyond the typical 2100. And the reason we're interested in this is that some of the reservoirs in the ocean that have overturning and renewal timescales that are longer than a century could have interesting dynamics in terms of how they play out past the year 2100. Okay, next slide, please. So with this question of nutrients and looking at the lower trophic level behavior, I want to just start by describing and reminding people if they have seen this before, the sort of very influential work of Jorge Sarmiento from 2004, who unfortunately passed away just a couple of months ago. Anyway, he had this very important paper in 2004 where he presented a view of global biogeochemical cycles in a sense, a framework. And the study had two main parts. The first part shown on the left is a modeling study, a sensitivity study they did, and the right is their kind of part of their attribution, their preferred pathway for how things are working on global scales. Let me just emphasize on the left, what Sarmiento et al. Did was they took a model and they asked the question, how much influence does the Southern Ocean surface nutrient field have on global productivity over the other regions of the ocean. So they did a model simulation with their full biogeochemistry model, and they did another where they zeroed surface nutrients. And what they found in the model, it's kind of shown here on the left, but I'll explain it, is that over the low latitudes, so not the Southern Ocean, but the low latitudes, remotely, 75% of the productivity is controlled by the export of preformed phosphate from the Southern Ocean. And this is the figure on the right is their preferred subantarctic mode water and interior water mass. We don't need to go into the details here, but they argued that this has global reach, and this is kind of a mapping of how the water is moving around. So key idea, Southern Ocean controls large-scale biogeochemistry. So next, please. And this was followed up by a study by Moore et al. That was published in 2018 in Science. And whereas Sarmiento's study was with a steady state model, did not have a perturbation, this used a future projection at 2300, the CESM model. And what these people in this study showed, what they argued was that with the CESM model under a strong forcing, as RCP 8.5 forcing, that nutrient trapping, so in this figure, the bottom left figure is phosphate zonally averaged. And nutrient trapping, according to their interpretation in the Southern Ocean, by this Sarmiento mechanism, results in a depletion in the future relative to the present of phosphate in the low latitudes. The Southern Ocean just stops exporting phosphate to the rest of the ocean, and you have productivity drops of order 30%. And NPPN export drops by of order 30% in the low latitudes remotely in response. But they didn't actually test this by looking at transports, or it was just assuming that the Sarmiento mechanism was operating. So we can go to the next slide, please. And so what we did in our Nature paper was we looked at the question of whether low latitude regenerated nutrients may be, in fact, playing a more important role than others had thought before in terms of sustaining low latitude productivity, and that the Southern Ocean nutrient mechanism of Sarmiento may be part of the story, but certainly not the whole thing. So I won't go through this in too much detail, but the left panel is just on a thermocline and on a subantarctic mode water density level phosphate from observations, and then a section right up down P16 right up and down the Pacific on the bottom left. And then on the right, using an AOU deconvolution, the regenerated fraction shows the regenerated fraction from observations in these different depth horizons that is regenerated, that is due to remineralization. And throughout the low latitudes at different depths, it's of order 50% or more. So if we can go to the next slide, then the main inference from observations that there's a very large remineralized pool in the low latitude mesopelagic domain, and it essentially can interact with a southern ocean supply for promoting primary productivity. Sort of the question we were asking. And then, but not, it's not absolutely clear, where does this remineralized pool in the low latitudes come from? Is it locally regenerated or from the Southern Ocean? And so if we can go to the next slide, we looked at some mechanisms. We did a mechanistic perturbation study that was analogous to what Sarmiento had done in the Southern Ocean, but this time by looking at low latitude nutrient supply. So the left column shows the primary production, the right column export at 100 meters. So the top row is just for this. This is with the Nemo-Piscus model run in a steady state configuration like what Sarmiento had. And the top row is for primary production and export for the control run. Then we did not allow in the mesopelagic domain remineralization for the middle for this pert trop for our own perturbation study. And we found, and then the difference between those is shown at the bottom. So if we look at the text at the bottom right side of the slide, we can see that when we disallow low latitude remineralization, we have a 72% drop in primary production and a 55% drop in export. And this is actually quite remarkable because these are numbers that are at least as large as what Sarmiento had identified in a much older generation model. And so we interpreted this to mean that low latitude remineralization locally is in fact critically important for primary production. We can go to the next slide, please. Thank you. And then we in the final part of our paper, we just showed some results from CMIP 6 models that were run out to 2300, showing global upper left is global primary production fractional changes, and then the fractional changes over 30 south to 30 north for the for the 5 models that we had that were done to 2300. And you see this wide range of responses. They disagree in both the amplitude and sign. And what we argued in the paper from the analysis shown in the bottom row is that a lot of this difference can stem from whether a model has temperature-dependent remineralization or not. So the model with the strongest positive response, the IPSL CM6A model, you can see in the lower left panel, has an increase in phosphate throughout the upper ocean. This is a zonal mean over the upper 800 meters. The mesopelagic domain in general has an increase in phosphate. And a model that the CESM2 model, which does not have temperature dependent remineralization, does not respond to temperature perturbations for remineralization rates, shows a loss. And that is one of the models with a substantial, with an important decrease in primary production. So we argued in this paper that this kind of mechanistic uncertainty could be important for understanding this wild disparity between Earth system model behavior. And then if we can go to the next slide, please. Thank you. So there's a little bit of detail in here, and I think for the FishMIP people, it's not necessary to communicate this, but I'll just go over it quickly. So we reached out to marine ecosystem modeling groups, and through FishMIP, to which Daniele Bianchi is a participating member, and we considered runs with ESMs that were done to 2300 under 3 scenarios: high emissions, low emissions, um, and an overshoot scenario. So, and we were very interested in this question of what is the gain of climate mitigation? And I'll come back to the uncertainty question later that I was just describing for the ESMs themselves. But we were interested in getting a little bit into the story of what controls uncertainty with these projections. So we can go to the next slide, please. So as a matter of context, I just wanted to highlight this is from a study from this SROC study from 2019. I believe this figure was made by Alessandro Tagliabue, and the showing under this is out to 2100 under RCP 2.6 and RCP 8.5. And you can see the changes. Stippling indicates where there's the models are uncertain in the sign, but the models do not agree in the sign. But over for both the high emission scenario lower right and low emission scenario lower left, for fish biomass, fractional fish biomass changes, the uncertainty looks quite small here. So you could argue under, for instance, RCP 2.6, under this 2-degree stabilization, the models are reliable. They have fidelity to each other in terms of their behavior under this kind of 2-degree stabilization. So this can be used for the models are useful for investigating impacts. So we can go to the next slide, please. And I think everyone is probably familiar with this slide from Tensor from 2021 that shows uncertainty compared across the models, and but it doesn't do a systematic investigation of it doesn't do a systematic investigation analogous to what Tagliabue had presented in the SROC report in terms of maps with uncertainty. So this is just to give us an idea of where we're going. Can we go to the next slide, please? Okay, this slide is a little bit of a lot of information, but I want to just emphasize that in these runs to 2300, we are comparing, we are interested in using 3 Earth system models: IPSL CM6A, UKESM1, CESM2. And in addition to that, six FishMIP models. So each of these models is run, they're effectively 3 times 6 is 18 iterations with each scenario. And there are 3 scenarios. So this gives you an idea of what we're using here. I won't go into the details. Just remember they're 18. If we can go to the next slide, please. Just in the averaging across the models, this is a view of just the response. So biomass changes, SSP 585, which you can see is the one with the strongest drop in normalized biomass. decreases by of order 50% under approximately 10-degree warming. SSP 126 and the overshoot SSP 534 have approximately 10% loss. And the figure on the right shows by 2300. The figure on the right shows the biomass loss, fractional biomass loss against atmospheric CO2. And it's interesting, this shows not quite linear, but linear-like, I guess you could say. And you can see a little bit of an element of this overshoot with SSP 534 in both figures, where there's a little bit stronger response, but it more than rebounds by 2300. Okay, next slide, please. Now we're going to just go through and we're going to, now that didn't include any uncertainty information. Now we're going to go through with the uncertainty information and say that, look at these figures on the left, and we have SSP 126 on the upper left panel showing global fractional biomass change, where we've included not only the ensemble mean as in the previous figure, but also uncertainty separated by the contribution from ESMs and from marine ecosystem models. And under SSP 585, the second down on the left, we can see this decrease to 50%, and we can see the uncertainty growing in time, and it's dominated by the marine ecosystem models. But in both SSP 126 and SSP 585 over global scales, the uncertainty does not cross zero by 2300. It does not span zero. So this is a significant decrease in both cases. Okay, and then for the mitigation in the lower left panel, oops, for the gain of mitigation in the lower left panel, remember this is SSP 126 minus SSP 585, so it's positive, and this goes up to about 40% as the difference. And this, we can interpret this to mean that by having low emissions relative to high emissions, we have a 40% mitigation gain in fish biomass, fractional fish biomass loss. Okay, next slide, please. Okay, so what do the patterns look like for this? So let's first look at the upper left panel. For SSP 126, we can see that in colors over large scales, they're ignoring the Arctic and the Antarctic regions, we can see this bluish 10% loss that's relatively not too structured with some differences over large scales. But importantly, at the 90% confidence level, there's stippling over most regions, which means that the forced change, the forced anthropogenic change, is smaller in this representation of stippling than the intermodal uncertainty. So on regional scales, even at 2-degree stabilization, the models, if we do this, it's more of a Hawkins and Sutton. It's just a more statistical analysis of the amplitude rather than the sign. We have this somewhat concerning result that we don't have many regions where the signal is emergent, so to speak. SSP 585 is in the next panel, the middle panel, and there we can see, because the force signal is larger, that the signal to noise ratio does emerge in some regions over approximately half of the global domain, and we see this approximately 50% loss over most of the ocean. And for the gain, we can see that, remember it's of order 40%, we can see, like SSP 585, this has emergent behavior over most of the globe, or of order half the globe. And so this is giving us a view of how robust, given model uncertainty, and maybe considering some of the mechanisms I mentioned before, the response can be. Okay, if we can go to the next slide. This I'll just go over very quickly. This is for the contribution of what is the relative. We'll just focus on the upper right and lower left panel here, and it shows how much of the uncertainty is contributed from ESMs, upper right, and emphasizing the low latitudes for ESMs in the upper right panel, and the Southern Ocean and the high latitudes of the Northern Hemisphere for marine ecosystems in the lower left panel. So there are different regions over which the two types of models are contributing uncertainty. And we could only do this because we're running the groups that are doing this are running the marine ecosystem models offline. So it allows us to do this partitioning. It's very useful. And natural variability is not contributing very much from the lower right panel. If we can go to the next panel, please. I probably have to hurry because of time constraints here. And so this shows a focus on large marine ecosystems. And this is going back to the gain, emphasizing the gain of mitigation, defined as the difference between the SSP 126 and SSP 585. What is the advantage that's brought by strong mitigation relative to high emissions? And this is ranked by the ensemble mean for each case in the 2290s. And you can see that many of the models are up between 50 and 40%. Many of the large marine ecosystems have gain of order 50, 40, 30%. And where the uncertainty bars do not intersect with zero, we can consider those to be a robust expression over this longer time scale of mitigation gain above the uncertainty level. And so we can see, I guess since I'm located in Japan, I was first looking at the Kuroshio current, which is maybe the sixth or seventh down, and a couple down is the Oyashio current, and they're both clearly robustly emergent. But when we get down to some of the sea ice covered regions and also some of the low latitude, like in the Indonesian Strait region, um, we can have very, very large uncertainty that can obscure mitigation gain. And so I won't go into much more detail unless there are questions about this, but we get for approximately uh, half or a little bit more of the global large marine ecosystem domains, we have a robust emergence of mitigation gain, which we don't have looking at SSP 126. So it's interesting to consider, are these perhaps both valuable ways to look uh, at impacts and to understand what the degree to which models can be used to argue for the value of mitigation. Okay, and the next slide, please, is the final slide. Great, thanks. So in conclusion, for fish biomass projections to 2300 using 3 ESMs and 6 marine ecosystem models, we see robust decreases in fish biomass at 10% under stabilization 2 degrees for SSP 126 and up to 50% losses under SSP 585, which is really very, very large losses. And the gain is robust at 40% when considered over global scales, and it does have some structure, and it does have important uncertainty itself. So the uncertainties for the case of SSP 126 are concerning as it potentially limits applications to adaptation and underscoring the need for improved mechanistic understanding. And so there's a lot of work to be done to really get at the sources of uncertainty in the different types of models and to hopefully move towards a more robust emergence for the 2 degree stabilization SSP 126 type scenario. And the final comment is that biogeochemical parameterizations in ESMs are an important source of uncertainty for SSP 126. And it comes back around to this idea that remineralization does seem to be particularly important, so that we're doing some work with Laurent Bopp, with the IPSL model, and trying to understand through sensitivity studies with their modeling framework, which we will then hopefully interface with fish modeling groups to test in a way specific sensitivities that can't be so easily evaluated just using ESMs. Okay, so that's it. Thank you very much.
Thank you very much, Keith. That was a super interesting presentation. We have time for questions now. Please don't forget to write your questions on the Q&A box at the bottom of the panel. I'm sure the questions are streamed quickly. Keith, I just have a question overall about like next steps and what other like experiments or work the working group is planning. I mean, you mentioned some work to understand with Lauren Bob to understand parameterizations better. Is there other plans to do like other comparative studies?
Yes, that's a great question. So Laurent Bopp has set up and hopefully this can open a broader discussion. He set up a offline configuration of his of their Pisca's biogeochemistry model. And so they can force it with the output from a CMIP6, the physical components of a CMIP6 perturbation, of CMIP6 simulation, or CMIP7 simulation, and then they can run the model offline with exactly the same circulation and warming and everything, and they can perturb different parameters, and then use the output of that to drive fish model, uh, marine ecosystem models. So I think the idea is to open a discussion between the Earth system modelers and the biogeochemical modelers, the marine ecosystem model people and the Earth system modeling people, to really draw on both to coordinate perturbation studies that are informed by the great learning that people have already made. I'm new to the marine ecosystem modeling part, so it would be great to open up a discussion if that answers your question.
Yes, it does. Thank you. Let me see if there are any other questions on the Q&A box. Okay, I think on the Q&A box under the more button of the screen. I don't see any questions for the moment. Okay, there's one right there. Can you see it? Okay.
Yes. Oh, there's a question about the biomass increases in the Arctic. So that's a great question. The Arctic is in those maps that we're showing, for SSP, say for SSP 585, 50% reduction in biomass in the non-ice covered regions. This gets back to the idea of with ice retreat, that you can have, for example, with the stemming from ESM behavior, you can have responses such as a breaking the iron limitation constraint. So you can have very model dependent wild primary production and primary productivity and biomass increases in those regions as ice retreats. And there's a fish biomass response that is in accordance with that. So that's the anomalously positive. That's where there's gain. But and I am not certainly that experienced with these marine ecosystem models at this point, but Daniele Bianchi has cautioned me that in FishMIP culture, that the high latitude ice covered regions are usually described, if I can quote him, as being areas where, we're still working on that, and that there's not the same degree of confidence. I may stand corrected on saying that, and I may have misquoted Daniele, but I think that's kind of the gist of what he told me.
Thank you so much, Keith. I don't see any other questions for the moment, but we will have time after Gaël's presentation to continue with more questions. Thank you again. We're going to move to our second presenter, who is Gaël Mariani. Gaël is a postdoctoral researcher in marine ecology at the University of Montpellier in France. He got his PhD in functional ecology in December 2023 from the University of Montpellier. And he studied the contribution of fish to ocean carbon cycle and the potential impacts of climate change and fishing on this contribution. Gaël, over to you. You have 20 minutes for your presentation.
Thank you, everybody. I have to put my mic on. Good morning and good afternoon, everyone. I'm going to present my work made during my PhD at the University of Montpellier, whose aim was to estimate the role of fish in the carbon cycle. Next slide, please. So today we know that the ocean absorbs a huge fraction, 30% of anthropogenic CO2 emissions, and stored about 38,000 billion tons of carbon. And compared to the atmosphere, it's a huge stock of carbon. And among the processes that promote carbon storage in the ocean, they are biological processes. Next slide, please. Biological processes, they are all the processes that enable carbon to be transported from the surface waters to the deep sea. This is what we call the biological carbon pump. And for a long time, we thought that planktonic organisms, so phytoplankton and zooplankton, had a huge contribution to the biological carbon pump. But more recently, next slide, please. More recently, we started to understand how hygrotrophic levels, including fish and squid, contribute to the biological carbon pump. And there are several pathways by which big individuals, big animals contribute to the carbon cycle. If we take the first pathway, fish produce fast-sinking fecal pellets, and the carbon contained in the fecal pellets can sink to the deep ocean. The same for the carcasses. Dead carcasses can sink to the sea bottom and sequester the carbon that is stored in the dead carcasses. We also have vertical migration performed by mesopelagic fishes. They feed in surface waters and defecate and respirate in mesopelagic waters, so the carbon is sequestered. And there are also indirect mechanisms, such as the mechanism number 4, where fish pee can provide nutrients that boost primary productivity and, at the end, carbon export to the deep sea. On the coast, we have some papers showing that big predators and high trophic level species are able to threaten fish that feed on seagrasses, for example. So the presence of high trophic level reduce herbivory and increase carbon sequestration in a blue carbon habitat on the coast. So today we have some proof that fish directly and indirectly contribute to the carbon cycle. Next slide, please. The question we can ask now is whether this role in the carbon cycle is under threats. Under threats because of climate change. We know today that climate change will lead to a decrease in 20% of the biomass of hygrotrophic levels. And under threat because of fishing. We know that every year around 90 million tons of carbon of fish are removed from the ocean, and some papers suggest that fishing has reduced the biomass of fish by 32% compared to the pre-industrial level. So the main goal of the PhD thesis was to assess what was the past, what is the current, and what will be the future contribution of fish to the ocean carbon cycle in a climate change and fishing context. Next slide, please. To do that, we used the model developed by, partly developed by Daniele Bianchi, the boats model. And we were able to estimate fish biomass, so it's only commercial fish biomass, not accounting for the mesopelagic fish, for example. We were able to estimate fish biomass for the period of time running from 1950 to 2100 under two climate change scenarios, SSP1 2.6 and SSP5 8.5. And we also run two contrasting fishing scenarios, one without fishing and one with historical fishing until 2014, and the same level of fishing until the end of the century. We also estimated carbon fluxes from sinking dead carcasses using a mortality function that is temperature and mass dependent. And we estimated carbon fluxes and export from fecal pellet production using, again, a function that is temperature and mass dependent. Next slide, please. We then estimated carbon export toward the deep sea using a carbon export model accounting for bacterial activity that is able to remineralize the organic carbon into carbon dioxide. And we also took into account two different sinking speeds for fecal pellets and dead carcasses. Next slide, please. And finally, we used an ocean circulation inverse model to see how and to estimate the time of sequestration of the injected carbon in the injected CO2 in the deep sea. So we estimated the time of sequestration as well as the fish carbon stock in the global ocean. Next slide, please. If we look at our first result, so the impact of climate change of fish biomass and carbon fluxes and export, we can see that the biomass is expected to decrease by 24% by the end of the century. And we can also see that the decrease is smaller for fecal pellet production and the sinking of the dead carcasses, because by the end of the century, we have a decrease that is around 13.3% compared to the historical period. Next slide, please. Another way to see those results is by looking at the change in carbon here on the y-axis as a function of the change in atmospherical temperature. And when we look at the slope for biomass, so the left plot, the slope equals minus 4.3%, meaning that every 1 degree increase in temperature, the biomass will decrease by 4.3%. And on the right plot, we can see that the slope is lower, minus 2.5%, meaning that carbon fluxes are expected to decrease by 2.5% every 1 degree increase in temperature. Next slide, please. And this result can be explained by how the model takes into account different theories, because the first thing that we can expect is that an increase in temperature will lead to a decrease in biomass, a decrease in fish consumption, and therefore a decrease in fecal pellet production. But there are several rules or theory saying that an increase in temperature leads to a shrinking in fish size. And this shrinking in fish size combined to the metabolic theory of ecology, the metabolic theory of ecology saying that there is an hypoallometric relationship between consumption and fish size, leads to an increase in fish consumption at the same level of biomass. So an increase in consumption can also mean an increase in fecal pellet production, and therefore there is a little compensation compared to the loss in biomass. Next slide, please. And there is another factor that is taken into account in the model. This factor is the fact that an increase in temperature will drive an increase in fish consumption, because fish needs more energy when the temperature are higher, and it will also compensate a little bit for the decline in biomass. Next slide, please. And next slide again, please. If we look at the impact of fishing now, on the left panels, you have the impact of climate change only, and on the right panels, you have the combined impact of fishing and climate change. And we can see that the historical impact of fishing is far more important than the historical and future impact of climate change, with a decline in biomass around 71%, that's only for commercial fish, and a decline in fish fecal pellet production and fish carcasses of 47% and 62% respectively. So when we see these results, one might ask if it can be worth it to restore fish population to sequester more carbon. Next slide, please. Another way to ask this question is to understand, estimate if fish stock restoration can be a new natural climate solution. A natural climate solution, it's a conservation, restoration, or ecosystem management action that mitigate climate change by promoting carbon sequestration or avoiding CO2 emissions. And more than mitigating climate change, it should also benefit people and biodiversity. And the current natural climate solutions, all of them are on land or coastal areas, are expected to mitigate climate change at a level of 10 gigaton of carbon dioxide per year. So here we will see what can be the result if we take into account the ocean and fish. Next slide, please. So to do that, we will answer the first question: does restoring fish stock can have an impact on climate? The same for biodiversity and the same on a society. Next slide, please. For the climate, we first have to show that fish stock restoration can impact the ocean carbon cycle, and then we have to show that fish stock restoration impacts atmospheric CO2 concentration. Next slide, please. If we look at the impact on the ocean carbon cycle, our results suggest that if we are able to restore fish stocks at their pre-industrial level, the maximum carbon sequestration potential of these measures would be around 0.4 gigaton of CO2 every year. Next slide, please. If we compare fish stock restoration measure to the maximum sequestration potential of other terrestrial and coastal measures, we can see that for the maximum potential of forest restoration measure is an order of magnitude higher than the one of fish stock restoration. But between fish stocks measure and mangrove restoration measures, that's the same order of magnitude. Next slide, please. What's interesting to look at also is the time of sequestration, because when fish poo and when the fecal pellets sink to the deep ocean, the carbon contained in it is sequestered for a big amount of time, around 600 years. And it's most of the time higher than the time of sequestration in other ecosystems. So that's something that we can take into account, because the carbon sequestration is more permanent compared to other ecosystems. Next slide, please. So restoring fish stocks might have an impact and will probably have an impact on the ocean carbon cycle. What we still don't know if it has an impact on atmospheric CO2 concentrations. Next slide, please. And it's kind of difficult to estimate, because if we are in a scenario like today, where there is a lot of fishing and where fish stock are lower, and that we reduce the fishing to increase fish stock, we have to prove that increasing fish stocks increase carbon export to the deep sea, but also, next slide, please, but also increase the uptake of CO2 from the atmosphere to the ocean surface. And this kind of calculation, that's something that we were not able to do, and we absolutely have to do it before saying that restoring fish stock can meaningfully impact CO2 concentration in the atmosphere. Next slide, please. And because it's difficult to track the exchange of CO2 between the atmosphere and the ocean, some researchers argue that natural carbon uptake by ocean biology, which include restoring fish stock population and restoring the capacity of fish to sequester carbon, will not deliver credible carbon credit. So we have to be cautious because today we are not able to say that fish carbon can help us fighting climate change. Next slide, please. So we still have to study it. Next slide, please. Now, if we look at the impact on biodiversity, obviously restoring fish stock population will have a positive impact on biodiversity, especially because it will restore fish stock and commercial fish stocks. But we can try to think about indirect positive and negative impacts of restoring fish stocks. Next slide, please. And among the positive impacts, we can think about deep sea scavengers, because before the pre-industrial period, we can imagine that scavengers in the deep sea were feeding on dead carcasses coming from the epipelagic realm. But today, and our result shows that there is a decrease of carcass arrival by commercial fish of 60% compared to the pre-industrial level. So we can imagine that if we restore fish stock population, it will increase the sinking of dead carcasses and provide more food to deep sea scavengers. There might have also some negative impact on deep sea biodiversity, because if we want to increase biomass of fish in the epipelagic ocean to increase carbon fluxes and carbon export, it will also increase the remineralization of carbon in the deep ocean, which can cause an increase in CO2 concentration in the deep ocean, so an acidification of the water, and it can also decrease the concentration in dioxygen because of the activity of bacteria. So next slide, please. Obviously, the positive impact are bigger, but we also have to think about potential smaller but negative impacts. Next slide, please. And now we will try to see what could be the impact on the society by estimating by seeing how restoring fish stock for carbon purposes can be in conflict with fisheries. Next slide, please. Because now it's quite clear that fish contribute to the ocean carbon cycle, but we also know that fish and by extension fishing provides a lot of jobs around the world and contribute to food security by bringing protein in the diet and micronutrients that are essential for being in good health. Next slide, please. The problem is that if we want to increase carbon sequestration in the ocean, it can impact negatively fishing. So one of the aims of my PhD was to estimate where are the potential conflicting areas between carbon sequestration here on the y-axis and fishing effort here on the x-axis. And if we look at the top right corner, this is the areas where we have both high fishing effort and high contribution of fish to carbon sequestration. And this is what we can call the potential conflicting areas. Next slide, please. And when we map these potential conflicting areas, we obtain this kind of map, and the result is that 11% of the ocean area is a conflicting area between carbon sequestration and fisheries. It means that in these areas, if we want to increase fishing, it will probably decrease the contribution of fish to the carbon cycle. And the opposite is also true. If we want to increase carbon sequestration by fish, it will probably decrease the fishing, the benefits of fishing. Next slide, please. So what we suggested in our paper is to use a case-by-case approach to minimize conflict, because, for example, if we take the potential conflicting areas on the coast, and especially on the coast where an high percent of the population is at risk of food insecurity, it's not a good idea to reduce fishery for carbon purposes. So the goal is not to increase carbon sequestration in this area, because it can have negative impact on population and on society. But on the other side, 60% of the potential conflicting areas are identified in the high seas. And we know that fishing in the high seas contribute only a little to the global food security. We also know that fishing in the high seas is associated with forced labor, that fishing in the high seas is not economically viable, and that fishing in the high seas can affect the economic viability of small-scale fishery. So we don't have an answer to the next question, but we can ask whether stopping fishing in the high seas can bring benefits for biodiversity, people, but also for carbon. Next slide, please. So if we summarize all the results and try to answer, can fish stock restoration measures can be new natural climate solutions, we saw that they are mostly benefic impact and positive impact on biodiversity, that we can have positive impact on society, but we have to make sure that on coastal areas there are no negative impacts. And we still have to study the impact of fish restoration measure on the atmospheric CO2 concentration. So for now, there are too many unknown, and the most important one being the atmospheric CO2 concentration, to conclude positively that fish stock restoration can be a new natural climate solution. Next slide, please. So that's it. Thank you for listening, and thank you to all the co-authors of the papers that helped me during my PhD thesis.
Thank you so much, Gaël. That was a super interesting presentation. We have a couple of questions for you in the Q&A box. I will start reading the question for Keith Friedman. It says, if we cannot set fish stocks will affect future climate, is the same true for mangroves and seagrass?
No, it's not exactly the same, especially for mangroves, because for mangroves, it's easier to track when the atmospheric CO2 goes to the mangrove biomass. So there are a lot of uncertainties on how restoring fish stocks can impact the climate, but it's easy to track what's the becoming of the carbon. What is more difficult for a mangroves and seagrass ecosystem is to estimate the impact of restoration measure on other greenhouse gases, such as methane, that because mangroves and seagrass might emit some of these greenhouse gases. So it's not as difficult for mangroves and seagrasses, but there are still some uncertainties.
Thanks, Gaël. There's another question from Christine. Christine is asking, do you have estimates of the contribution that mesopelagic fishes make to the BCP She's seen some estimates that they might be up to 90%, but not sure of the variability or uncertainty in that estimate.
There are some estimate of the contribution of mesopelagic fish. I don't have the number in mind, but it's higher. The contribution of mesopelagic fish is higher than the contribution of fish of commercial interest. And the biggest issue we have with I think it's 1 gigaton of carbon per year that is exported to the deep sea thanks to mesopelagic fish, but we still know a little about the biomass of mesopelagic fish. So that's the biggest uncertainty. And if we have a lot of uncertainty regarding the biomass of fish, of mesopelagic fish, the estimate of their contribution is also highly uncertain.
Thanks, Gaël. There's one more question. Connor is asking, do you think there could be a trade-off between fish and zooplankton-mediated carbon sequestration? For example,
yes.
if fish biomass, okay.
Yeah,
okay.
go ahead, finish. Sorry.
For example, if fish biomass decreases and predation on zooplankton is reduced as a result, we could imagine how increased zooplankton biomass might offset loss of fish-mediated carbon export. Do your analysis provide insights?
So it goes with Olivier's question, the last question. That's a good question, and that's something we were not able to take into account with boats, because boats don't take into account trophic relationship between fish and also between fish and lower trophic levels. So this is true if we increase fish stock. The predation on a lower trophic level that also contribute to the carbon cycle will increase, and therefore their contribution to the carbon cycle will decrease. So that's something we still have to do, taking into account the top-down impact of adding big fish or removing big fish on the entire carbon cycle and the contribution of all organisms to the biological carbon pump.
Thanks, Gaël. So I think you answered Olivier's question too.
Yes, I think so.
Yeah, at the same time, and I think we're getting close to the end of the webinar. So I just want to thank you and Keith again for your great presentations. Yeah, they were really nice. We really enjoyed them. And thank you again all for joining. The webinar recording will be available on the FishSCORE website, and there's also the past webinars are available there, the recordings. And our next webinar will be in late July, as Sinclair has posted the link to the FishSCORE website where you will be able to find a recording of this webinar. Thank you again all for joining, and hopefully we'll see you on the next webinar.